Good morning, everyone. I'm Rahab Yassin from the NCI Division of Cancer Biology, and it's my pleasure to start the first session in this symposium on mutation signature in normal and cancer tissue. We have 4 presentations scheduled for the session, followed by a moderated discussion. Each presentation is 20 minutes, so I'm going to play the role of the timekeeper. If the presenter is not wrapping up at 20 minutes, I will be interjecting with the time announcement. We have a rich agenda and hope to accomplish the goal of the day. So without further delay, it's my pleasure to introduce the first speaker for this session, Dr. Phil Jones from MRC Cancer Unit, University of Cambridge. And the title of his presentation is Environment, Selection, and Signatures in Squamous Epithelia. Dr. Jones.
Sorry for that delay. I'm Phil Jones. I work at the Sanger Institute and the University of Cambridge, and I'm a clinician, and I treat cancers of the skin, and I've also treated cancer of the esophagus. And the problem with these cancers, as with many cancers, is by the time we diagnose them, they already have complex, highly evolved genomes. And that problem means that they will resist treatment. So about 20 years ago, I started wondering whether if we understood the clonal dynamics of mutant cells in normal tissues, whether we could intervene much earlier and potentially prevent those much simpler mutant cells from developing into tumors and then prevent cancer before it starts. So that was the motivation behind the research I'm going to show you today. The beauty of these tissues is their uniformity. Some people would call them boring, but I think they're beautifully uniform. And they have a very simple structure. So the lower layers of the tissue contain all the proliferating cells. And then when cells commit to differentiate, they leave the bottommost layer and start migrating towards the tissue surface. And then at the surface, they're shed. And there's a continual process of cell shedding from the cell tissue surface and a continual process throughout life of proliferation to replace the cells that are shed. Now, the structure of the tissue is very different from the liver that we were just hearing about, because critically, there is no barrier to prevent the lateral spread of mutant clones. And as I'm about to show you, this means mutant clones can get millimeter or centimeter scale in size. And that is critical if you're going to understand the basis of clonal evolution and selection in these tissues. So Peter's shown images like this, and this is why we started with these tissues, doing low resolution maps. So if you could see in the mirror in your eyelid and your middle-aged or over 1 square centimeter of skin, you would see something like the image on the left here. The colored circles are genes under positive genetic selection, so with an excess of protein altering over silent mutations, which is our metric for defining a gene that's driving clonal expansion. And you can see that 20 to 30% of normal skin by middle age is colonized by genes that are driving clonal expansions, and these same genes are occurring at high prevalence in keratinocyte cancers of the skin. If you consider an internal squamous epithelium, also with a similar structure, that of the squamous esophagus, actually the picture looks even more dramatic. The same genes are under selection, but they seem to have colonized even more of the tissue. So these are both top-down views generated by ultra-deep targeted sequencing. There's still quite a lot of white space. So if you zoom in and you do micro mapping with tiny punch biopsies, and the scale here is much smaller. This is normal skin from a 75-year-old shoulder. And these little holes are a sheet of skin where we've taken little punches. And the colored squares here are clonal whole genomes. And what what the trees here are showing you are independent clones coming from these colored squares. And the slightly worrying thing here is that you can see that we have multiple clones almost adjacent to each other, each of which is carrying multiple driver mutations. Some of them have copy number alterations. The number of substitutions across the genome is topping out in some cases over 40,000. So these are very mutated clones. Actually, if you look at the signatures, one group of clones here, this one here in pink, has a statistically different signature. And like Peter was saying with the liver, we don't know why, but you can have divergent signatures. They're all flavors of signature 7, but different signatures in this UV exposed tissue, despite being in close spatial proximity. So these squamous epithelia are very, very mutated. What's striking in terms of mutational signatures is these signatures and the mutational processes that underlie them are very different. UV, or in the case of the skin, in the esophagus, these clock-like cell-intrinsic SBS1 and 5 and intrinsic processes, different signatures, but very similar genes are being selected. We can go further than that, though. We can change the species, and we can change the mutagen and the time scale. So we can go from decades to months. We can take a mouse and look at the esophagus after 2 months treatment with diethyl nitrosamine, and I'm showing you the mutational spectrum here. And we can then sequence the esophagus with the same kind of protocol, deep targeted sequencing of a sheet of esophagus, and we get a very similar landscape. Again, there are more mutations here. The mutational burden is more like a 700-year-old human, but nevertheless, the same genes are under positive selection. But very nicely, because this is a mouse, we can then work out the clonal dynamics of what's happening by using genetic lineage tracing. This is a transgenic trick where we label cells, individual cells, with a yellow fluorescent neutral reporter. And in a non-mutagenized mouse, we can count lots of clones and then do the math and show that these are following neutral drift. So that's very nice. Constant proportion of labeled cells across a 1-year experiment. In the case of nitrosamine treated animals, you get savage clonal competition. You can see here that whereas in neutral drift, lots of little clones and a decreasing proportion of clones, the surviving clones are larger. Here, the number of clones has drastically reduced. The surviving clones are huge. So there is savage competitive selection going on here. But of course, because this is a mouse, again, we can cut out individual clones and sequence them. And what you find is interesting is that clone size is not simply a function of the mutations they carry. And it turns out that the size of clones depends on the neighbors of the clones. So what's happening is what we call a neighbor constraint model. And clones expand. This is a simulation, but it fits all of the data. And when they collide with an adjacent clone of similar fitness, then that clone with a proliferative advantage reverts back to neutrality. And that's how you can have clones with a strong proliferative advantage remaining in a normal tissue. So we have a simple tissue in which we can subvert neutral cell behavior with a mutation. And then once we've colonized the tissue, we revert back to neutrality and the tissue remains normal. And that's how those strongly selected genes are emerging through selection. So how can we use signatures to understand more about this landscape and its development? Well, we think about signatures in 2 ways. So they're very useful for environmental cues, understanding environmental agents that cause DNA damage, and those induce mutational signatures and new clones. But obviously, we can also have the same environmental agent causing clonal selection, and Peter talked about this in the last talk as well. But that agent might cause no mutational signature as well as expanding pre-existing clones. So let's start thinking about environment, and I'll talk a little bit about the skin. So this is a human ear, and we sequenced it using our grid mapping technique. So we're doing deep targeting sequencing over the inner and outer surface of the ear. And this is a beautiful illustration of the power of UV light. So the outer surface of the ear is tanned, and you can see in this heat map of the mutation of density of clones, the way that this shoots up the moment we get into the outer surface of the ear and into the tanned skin. So it's very clear that UV light has a huge effect on mutational density in the skin. In this study, there was one outlier who had a very high mutational burden in the skin. This was a transplant recipient receiving azathioprine, and their mutational signature was SBS32 azathioprine. And it turned out in this patient, they had very little UV. Their landscape was dominated by mutations generated by immunosuppression. And these patients have a very high incidence of squamous cancer of the skin. And this is suggesting that those cancers might be being generated by the drugs they're receiving to suppress their immunity. We've also looked across the body, and the reason we looked across the body is that there's a much higher incidence of keratinocyte cancers in sites commonly exposed to the sun, like the face, compared with other sites like the abdomen, where sun exposure is rare. And we see very nicely differences in mutational signature across the body. So you've got 7D in the face, and you don't find that in, say, truncal skin. And this is arguing there are differences in either damage and or repair in a site-specific way in human skin. What's interesting about that, though, is we also see differences in genetic selection across body sites. So again, in the face, in a human, P53 is more selected in facial skin. And here we're lining up mutations in facial skin against mutations in all other sites. And the gene should lie on the line. P53 does not. FAT1 does less well in facial skin. P53 does better. In the leg, intermittently sun exposed, we've got FAT1 doing better and Notch1 doing better. So where you are in the body determines which genes are being selected. And that's probably a function of exposure, frequency of exposure to UV lights, we think. And to prove the point, really, we've done a mouse model. And here, these green blobs are transgenically induced P53 mutant clones. And you're looking top down on sheets of skin. We've got adjacent non-UV exposed skin. And here we've exposed sub-sunburn level doses of UV light, the adjacent skin. And you can see these P53 mutant clones are massively expanded by low doses of UV light. So UV not only generates mutations, but it massively expands pre-existing clones. And the implication of this work, and this is an idea proposed by Doug Brash at Yale 20 plus years ago, is that most of the P53 mutant cells in the skin are really driven by the expansion of pre-existing clones growing rather than new mutations. And I think that's very important for the epidemiology of skin cancers. So our sort of what we hoped found in this looking across body sites, would we find an explanation for why the rate of skin cancer differs across the body. So that didn't really work. So then we looked across the globe, and we did this comparing the UK with Singapore, and this was done with Birgit Lane in Singapore, and this is work from Charlotte King. The UV index, how much UV gets to the surface of the Earth, is much higher in Singapore. There are differences in the germline if you compare Singapore and the UK, so positive selection in Singapore population for not only skin pigment genes, but also other gene loci that might protect against UV light exposure. But the really dramatic difference is when you look at skin cancer incidence. So the UK has a tsunami of keratinocyte cancers. Singapore, the incidence has hardly changed. It's 17-fold lower. So what Charlotte did was to construct a gridded array of epidermis from facial skin, from people from Singapore and the UK, from ultra deep targeted sequencing, map clones spanning multiple samples, and then collate the data. And here you can immediately see who is from Singapore with very few mutations per sample and who's from the UK with far more mutations per sample. We estimate the burden from synonymous mutations in the targeted data, and you can see that the UK people, I wish to say these are overall very similar in age in the two groups, have a much higher mutation burden. What was really interesting was the mutational spectrum, which was very different. And when we run SIG Profiler, you can see it's almost inverted. The Singapore mutations are, the majority of them are age, clock-like signatures, SBS 1 and 5. The minority are UV, and that's reversed in the UK. So that was a real surprise. Selection, again, let's plot non-synonymous mutations UK versus Singapore. And again, if everything's equal, everyone should lie on the line, but P53 is enriched, possibly selected in the UK, Notch 1 and Notch 2 in Singapore. Look at our maps and the difference becomes immediately apparent. There's a ton more P53 in this kind of orangey color in the UK and far fewer mutations in Singapore. Now it's very tempting, and I used to think this, that more clonal drivers as in positively selected mutations equals greater cancer risk. I just want to point out to you, I think that's a bit of a naive assumption of mine. So in UK skin, actually, there is no real increase in the prevalence of Notch1 and FAT1 in normal skin compared with keratinocyte cancer. There is for P53. And I put it to you that we really can't just define cancer drivers based on their prevalence in cancer. You have to compare cancer with normal. I think the esophagus also has a health warning in this regard. So look at Notch1 mutations in normal esophagus and compare it with cancer. Most people who are middle-aged and over on this call, the majority of your esophagus will have lost both copies of Notch1, one by mutation and one by LOH. And the reason that's happened is that when you have one mutated allele, it's haploinsufficient. There's a moderate increase in fitness and those clones outcompete wild type cells in your esophagus. When you use both alleles, it's hugely better competitor and Notch1 null clones take over your esophagus. And the reason that those don't give you cancer is that established large tumors need Notch1 to grow. And we've shown this genetically, but also with a function blocking antibody from Genentech. If you give the antibody, the Notch1 tumors in a mouse model, the tumors in the mouse model shrink. So you need wild type Notch to grow, and all your Notch1 null cells are protected from getting cancer. A second important comment on this is that very competitive clones in your normal epithelium can strangle and choke out and eliminate early tumors in the esophagus. So very briefly in summary, because I know I took too much time at the start, many factors shape normal tissue landscapes, and signatures give us a window to reveal some of these. How mutations relate to cancer risk, it's very clear they're both pro and anti-oncogenic mutations. I think we need to revise our definition of cancer drivers, frequency and cancer over frequency and normal. Really important. And the thing that we don't have and we really need is to identify clones in those transition states between normal and cancer. So, you know, we know in the esophagus, cancers have a ton of ApoBec and a vastly increased mutational burden. We haven't found in the esophagus, I think we found one clone out of hundreds that we've whole genome sequenced with ApoBec. It's really rare. It'd be great if we could understand those transition states and the signatures that they carry. At that point, I need to thank a huge number of people, and I'm going to stop.
Thank you very much, Dr. Jones. We have time for a couple of questions. I see some in the QA. I'm going to start in a random order. Great talk. Is there any copy number alteration in these normal tissue studies?
So it depends where you look. In the skin, there are most definitely a substantial number of clones with copy number alterations. When we do our sort of targeted approach, we find a substantial number. You can estimate that from SNP phasing, and there are probably 5 to 10% of clones with copy number alterations in the skin. In the esophagus, it's really interesting there that every large notch clone that we were able to look at has got copy neutral loss of notch 1, and every one that we've done whole genome sequencing where there's a notch mutation, you've lost the other allele of notch 1, and there really aren't many other changes at all in the esophagus. So the esophagus is very copy number genome stable, if you like. The skin has quite a lot of copy number alterations, but it deals with it, because I should stress this is completely histologically normal skin.
The next question, might UV also expand existing clones carrying BRAF mutations similar to the TP53 mutants?
So these are keratinocytes. So this is, BRAF is canonically a melanoma driver. If you look at gain of function, classical gain of function, your RASes and the like, you find them, they're really rare. They do occur. In the skin, PIK3CA and FGFR3 mutations are actually associated with benign tumors. So they seem to take cells in a different direction. And they're pretty rare in the esophagus as well. So I'm not saying they don't occur, but I'm saying they're rare.
Okay. And then we have a final question, or time for one more question. Are donors in the Singapore and UK of the same ethnicity?
They are not. I'm not sure I don't like the term ethnicity. They're not of the same germline genotype. Let's put it that way. So yeah, they differ in about 25 different genes under selection. So we were able to genotype them based off, and they do differ, particularly actually in one insertion in a gene called HILE2, which is said to be related to UV response. So I don't think the data is that good. And 4 out of 5 Singapore donors have this quite large HILE2 insertion. And that's very prevalent in people along the equator in South Asia, not just in Singapore. But it wasn't found at all in the UK. I think the germline is going to be very interesting, germline mutations, in looking at that. And it'll be really interesting to extend that to other countries, this study.
Thank you very much, Dr. Jones, for starting a rich discussion in this session. I'd like to remind everybody participating in this symposium, you can use the chat function to direct your questions to the presenter. We only have time for a couple of questions after each presentation. And with this, I'm glad to present our next presenter is Dr. Daniela Robles Espinosa from the International Laboratory for Human Genome Research, National Autonomous University of Mexico. The title of her presentation is Genomic Profiling of Acral Melanoma in Mexican Patients. Dr. Espinosa.
Thank you, Dr. Jason. Can you all now see my
yes.
presentation? Is that okay?
Yes, it's
awesome. Okay, thank you so much. So, good morning, afternoon, evening, everyone, wherever you're joining us from. Thanks to Ludmil, Tere, Ron, Mike, and the other organizers for the invitation to speak in this symposium, and you for joining this talk. My name is Daniela Robles. I am an assistant professor at the International Laboratory for Human Genome Research part of UNAM. And today I'd like to tell you about this project that we are, is very much under development in our lab at the moment, which is called genomic profiling of acral melanoma in Mexican patients. Now, this is a topic that we're interested in, and I'm going to tell you today what we're doing to learn more about it and how we're trying to use mutational signature analysis to help with our objectives. And as I suppose that there must be some students and perhaps people that are still learning about how to use these type of analysis in their work, I am hoping that this talk is informative about our experience. Okay, so here's an overview of what I'm going to talk about. First, I'd like to give you a brief introduction to acral indigenous melanoma, then tell you about what we're doing in our lab to tackle some questions about it, and then tell you a little bit about mutational signatures, what we've learned, and finally, a summary and some perspectives. Okay, so to begin with, melanoma has historically been classified in several clinical pathological subtypes, of which the 6 that you see here are the major ones. Now, these differ in their clinical presentation. They differ in their frequency in different populations, their prognosis, their etiology. For example, the first 3 here, superficial spreading, nodular melanoma, and lentigo malignant melanoma are associated with UV radiation, usually appearing in hair-bearing skin, whereas the next 3, mucosal, uveal, and acral indigenous, are thought not to be related to UV irradiation. Now, the topic of this talk, our focus is on acral indigenous melanoma. Now, its name, acral indigenous, refers to the location where it arises. Acral means palms of the hands, soles of the feet, and subungual locations, which means under the nails. And lentiginous refers to the shape of a lesion in the shape of a lentigo. Now, it is usually misdiagnosed as other conditions, at least in this part of the world, usually as diabetic ulcers or warts or fungal infections. And it is a rare subtype of melanoma in European descent populations, comprising approximately only 1 to 8% of diagnosis. However, if we look at the distribution of melanoma subtypes in select countries around the world, as I am showing you here in this map, then we see striking differences. Here, you see acral indigenous melanoma in red and the other subtypes I showed you before in other colors. And we can see here that it is the most common subtype here in Mexico, where I'm based, and in other countries such as Peru, some countries in Africa, and some countries in Asia. Now, an important note about this map is that it's only showing the proportion of melanoma subtypes around the world. It doesn't really tell us anything about the incidence. The incidence of acral indigenous melanoma seems to be the same, at least that's the best that we know at the moment. It seems to be the same around the world. And the differences you see here are thought to be due to differences in UV-induced melanoma in different places. But regardless, this is the most common subtype in Mexico and other countries in Latin America. And for us, this is an important reason to study it. We have other reasons also, because this type of melanoma has lower 5 and 10-year melanoma-specific survival rates than other types of melanoma. It's also understudied and its causes are currently unknown. So it is thought, as I said before, to be unrelated to UV exposure. But for example, some authors have plotted the locations of tumors in the soles of the feet, as you see here in this figure. And they have noticed an accumulation of tumors in the zones with mechanical pressure of the feet. So this points to perhaps mechanical pressure being an etiological factor. However, this has not been mechanistically proven yet, at least to the best of my knowledge. And the genetic landscape of acral melanoma risk is still very much under investigation. Now, it also has a different genomic profile to the UV-related subtypes. So as some of you may know, cutaneous melanoma related to sun or UV exposure is dominated by mutations in NBRAF, in NRAS, and the NF1 genes. The majority of tumors have mutations in these genes, about 85% of them. However, studies that have been published for acral melanoma show us that less than half of the tumors have these mutations, with possibly other drivers involved in the rest of the tumors. And finally, well, there is a lack of therapeutic options. Generally, immunotherapy seems to be a very good option for these patients. However, it is very expensive, and this is even more important here in Latin America, where the majority of cases are still treated with chemotherapy. Okay, so now I'd like to tell you about our project and what we're trying to do to investigate more about this cancer. And for this, we started this, though I like to call the Mexican Acral Lentiginous Melanoma Project. And this has been started in collaboration with institutes within the country. For example, the Institute for National Genomic Medicine, which is in Mexico City, and the National Cancer Institute of Mexico, and also international institutes such as the National Cancer Institute of Brazil and the Wellcome Sanger Institute. Now, before I go into telling you the data, I'd like to spend just a few moments telling you about the challenges of doing genomic research in low and middle income countries. And I know that maybe some of the attendees to this meeting will find that they are in this situation as well. So, first, there's not many studies of our populations available, and there have been papers and other studies that indicate that ethnicity plays a role in cancer risk and progression. So from our perspective, we need to start with a baseline, which means in some cases performing studies in our local populations that have already been done in other populations. And hopefully these will set the basis for knowing the genomic architecture of cancers here and allow us to just find results that are directly applicable to our populations. Now, also, there is a large disconnect between the medical and scientific communities here, and this makes large multicentric studies complicated in this part of the world, at least in my experience. Now, it is actually more expensive to carry out research here because not only are grants smaller, but also there are substantial import taxes for reagents and long waiting times for them to arrive. And finally, where there is a very large burden of bureaucracy and usually very little administrative and lab managerial support. So I'm just telling you all of this to put in context of, you know, sometimes what we have to deal in these parts of the world and why sometimes research goes a bit slower than in other places. But then nonetheless, we are working on this and our projects are trying to establish a baseline for the genomic studies in our populations. Okay. So with this in mind, the question that we have here is, what is the molecular profile, including driver genes, transcriptional features, and mutational signatures, characteristic of this type of cancer, acral lentiginous melanoma, in Latino populations? And for this, we started a collaboration with the National Cancer Institute of Mexico, also called INCAN, which is a reference hospital for cancer in this country, and the Regional Hospital for High Specialty of the Bajío, which is in the city of León, another central city here, to invite patients with this diagnosis to the study. Now I'm going to show you the patient characteristics at INCAN so you can get an idea of the population that we're targeting. So in this hospital, and in accordance with the map I showed you earlier, about half of the melanoma cases are diagnosed with the acral lentiginous subtype. And here you can see other characteristics about from these patients as well. So there's slightly more women diagnosed than men. Most of the patients come from the south of the country. This may just be because of the proximity of Mexico City to the States, as I'm here showing where Mexico City is. The majority of these patients arrive with a late stage of disease. Many of the tumors are already ulcerated in stage 3. A third of these patients have already died from their melanoma. The age of diagnosis is very broad, as you can see here, but the median is about 62 years of age. Tumors are already thick, according to a late stage when they are diagnosed, and the majority the primaries are diagnosed in the feet. There's some in the hands as well, and some patients already have metastases as well, mainly to the lung, to the brain, and to the liver. Okay, so when these patients go to a follow-up appointment, then we ask them if they want to join the study, and if they agree, then they provide a sample of saliva and tumor material in FFP form. This work has been coordinated by Christian Molina, who's a postdoc in the lab. And he's also coordinating a extensive manual curation of clinical information, as there's more than 100 clinical covariates recorded for each patient, which makes it sometimes hard work to keep everything clean. And finally, we perform exome and transcriptome sequencing from DNA and RNA extracted from the paraffin tissue and saliva. And we do this in collaboration with Simvestab Langeview, which is a sequencing center here in the country, and also the Wellcome Sanger Institute. Now some results, what do we have so far? So we have recruited more than 200 patients to this study, and this, to the best of our knowledge, comprises one of the largest cohorts of this type of cancer, at least that we're aware of. We have data for half of the tumors at the moment. Well, data analysis is still ongoing because of course we need to include the second batch of samples in our future results. And also we are doing signature analysis, and I'm gonna tell you about that in a moment. But first, I'd like to tell you a little bit about what we've learned. And this is work done by Patti Basurto, who's a PhD student in our lab. So what we see here from half of the data that we have already is that the mutational profile of acral melanoma Mexican patients does indicate that less than 50% of these tumors are classical driver lesions. Here with only 16% of these tumors having BRAF mutations, 13 and RAS, 14 KIT. We also see, as we have some patients that have more than one sample, that their mutational profiles are similar, but in some cases, of course, we find differences which may indicate important mutations that are, important for progression or for colonizing other tissues, for example, but this is still something that we're looking at. This looks similar to other landscapes published in other studies. I'm showing you two here examples from Australians and from US population, but we may have some potential differences with some genes here that we find maybe a bit more mutated than other genes in our cohorts, but we're still investigating this, and I still don't have a conclusion for this or if they are important or not, but we're still investigating this. And finally, we will need to perform a deeper analysis of these exomes when we have the rest of our cohort to hopefully identify novel gene drivers. And at the moment, yes, this is still work in progress, but it's what I can show you at the moment. Now, we have performed some preliminary copy number analysis as well. And we see genes that we already know of as amplified and deleted. For example, we see amplifications of telomerase, of cyclin D1, CDK4, and this novel gene called Crackle, identified by Richard White and MSKCC as a potential driver for acral melanoma, and deletions of genes such as CDKN2A and NF1. Of course, we see other peaks of deletions and amplifications where we have not found what gene or genes may be driving this, but we're confident that we have some good data, even if this is exome, to look deeper into this. And just one slide here about the RNA-seq analysis. This also is ongoing, but this suggests that there's some immunosuppressive microenvironment. The RNA reconvolution suggests that the majority of tumors are infiltrated by a high proportion of cells such as cancer-associated fibroblasts, to macrophages, endothelial cells, and regulatory T cells. And there's a higher infiltration of some of these cells in ulcerated samples and non-ulcerated samples. But I don't show you any graphs here because we need to corroborate these results with, for example, immunostaining images that we have that we are still doing. So, okay, but now the topic of this meeting and, what we've done about mutational signatures. So, as I told you at the beginning, we don't really know the causative agents for this type of tumor. And of course, that's where we think mutational signatures can be very useful and informative here to help us pinpoint potential causative agents. So this is our experience. So we tried running mutational signature analysis in our samples. This was with Thick Profiler Extractor, which is a tool written by Ludmila Alexandrov's group. And this was done by Caro Castañeda, PhD student in the lab. And this is the first thing we got, right? And you look at these, every column is a tumor, and every you see different colors as the proportion of mutations assigned to different signatures. And we see here, for example, some tumors seem to be dominated by mutations that have been associated with aging. Then we see this suspicious signature 3, which has been associated to defective hemoglobin recombination base repair, and we see a lot of that. Then we see 38, which is seems to be related to UV irradiation, an unknown etiology signature 40, and then a number of signatures that are related to UV. Now, we look at this and then we start thinking about, oh, we know what can this mean? What are the causative agents behind this? But the first thing we have to do is compare this with other similar studies, right? So when we compare what we got with another study of mutational signatures of whole genomes, so remember we have exomes, and this publication has whole genomes of acral melanoma in Australians, then we see, well, some signatures in common, for example, 1, 5, 40, 38, and the UV associated ones. They have more mutations, of course, mutational signatures because they have more resolution, and they are missing SVS3. So what is happening? This is our question. So this is something in discussion also with Ludmila and other people at the Sanger and other experts. This is what we've learned so far. And I'm hoping that perhaps this is useful for the students and people that are still learning about how to use this analysis. So first, at the moment, we have a collection of 100 exomes, which represents only about 2% of the genome. This means that we will have missed many mutation events in the genome, and we will have missed some patterns. For example, SVS13, which was very marked in the Australian study, we do not see that. Now, also, acral melanoma is characterized for having a very low mutational burden, and this may cause an incorrect assignment of mutational signatures. This is the case of SVS3. So SVS3 is known as a very flat signature, so it is very commonly miscalled. So if you get an SVS3 in your data, what you have to do is further tests to confirm if this is a real signature or it may have been miscalled. For example, by looking for genes that participate in the double break stand break repair. If they are missing or they have a deleterious mutation, then maybe your signature is correct. But if you don't do those analyses, this may be an incorrect assignment. Now, we also learned that maybe de novo extraction of mutational signatures may not be the strategy in this case, and it is perhaps better to combine or replace with signature fitting methods where you don't extract signatures from your data, but you compare to an already established catalog. And in this case, we have previous knowledge of what mutational signatures we may find because of, for example, the Australian study. And important questions there may be about if there are different proportions of these signatures and what the cause for that may be. And well, in some cases, as I showed you before, we have several samples per patient, in some cases primaries and metastases, but so as not to bias this analysis, it may be best to select only one for this type of analysis. So our preliminary conclusions from this mutational signature analysis exercise, well, we've learned something because both our study and the published literature show that UV radiation actually may play an etiological role in some of these acral melanomas, and these seem to be mainly those that are from subungual locations, meaning under the nails. So UV actually may play a role there, and we learned this from mutational signature analysis. Now, some preliminary findings need further proof to establish an etiological role, for example, SVS3, and we will need to also look at other mutational signature analysis tools, for example, indel copy numbers, to find more potential causative associations. Now, with this, I'd like to just give you a final slide of a summary and perspectives. So what I've told you today is that acral lentiginous melanoma is the most common melanoma subtype in Mexico and other countries in Latin America, in Asia and Africa, in some countries in this continent. Its causes are currently unknown, and at least for us in Latin America, there is a lack of therapeutic treatment for these patients. Our project, the Mexican ALM project, aims to sequence DNA and RNA and genotype these patients in order to search for genomic drivers and mutational signatures. So far, we can see that less than 50% of these tumors have classical driver mutations, that a large proportion of tumors we see the known oncogenes and tumor suppressors. However, there's still some peaks that we are investigating, and that they seem to be characterized by immunosuppressive microenvironment. Finally, mutational signatures can be very informative about the potential causative agents in cancer. However, as we've learned here, care must be taken when running the pipelines and a thorough review of these results is needed. And my last slide, well, here, what we want to do. Oh, sorry. Okay, that was my last slide. Sorry. Then I go to my acknowledgements slide here. And
thank you so much. Okay, I think there is a question here. Okay, so I don't see any question in the chat. I see one question in the QA. Have you seen any patterns of a mutation by anatomic location of the tumor?
This is a great question, and we're still, so as we have many more tumors at the moment from the feet, we still have no statistical power to confidently say something about this. However, it seems, we hope that we gain more power when we have our second batch of samples, but it seems that we may have more structural variants, for example, in the feet and in subungual locations than in the hands. That's what it seems so far, but we still have not enough power for that. But there may seem to be some differences there. Yes, thank you.
I see one more question being typed now. Do you see differences in mutational signatures between acral melanomas with non-driver mutation and without such mutations, without drivers?
We haven't done the signature analysis there, but we do see differences when we look at the structural variation landscape. The melanomas that have BRAF and NRAS mutations seem to have less structural variants, a lot less actually than the ones that do not have these driver mutations. So there seems to be a different genomic landscape, but we have not finished signature analysis yet. We're still working on that.
One final question from Dr. Gordinin. Were you able to look at index?
Yes, we do have some indel data. For example, we see some of those clustering NF1, for example, small indels at the moment. But because we have exomes, it's been a bit harder. We have called the copy number variants, but it has taken a lot of time. And what we want to do there, we're sequencing whole genomes soon. We still don't have this data, but we're hoping that that will give us more resolution to look at larger indels, because at the moment we only have data for very small, indels that are of very small size. But yes, we have some of that too.
Okay, thank you so much, Dr. Espinosa, for the presentation. We move now to our third presentation for this session, my NCI colleague, Dr. Maria Teresa Landi, and she will be presenting on landscape of mutational signatures in lung cancer. Teresa?
Yes, thank you, and to give me the opportunity to present our results on mutational signatures. So as you know, lung cancer is the second cancer type in cancer incidence and the first cause of cancer death in the world. And approximately 82% of the cancers are attributed to cigarette smoking, but the remaining are part of the never smokers, and never smokers have lung cancer occurring in quite higher frequency in certain populations, like in Asian populations, and is considered now the sixth cause of cancer death in the world. And only a few important risk factors have been identified in association with lung cancer in never smokers, but the large majority of the cases cannot be explained by these established risk factors, and this really highlights the importance to conduct more studies in the etiology of this tumor type. And this is the reason for which we designed the Sherlock Lung study. And the first goal of this study is to characterize the genomic and evolutionary landscape of lung cancer in never smokers. So the idea is to use a sort of a detective approach and using the tumor genomic changes as footprints or signatures to infer the etiological processes and also the evolutionary trajectories. So we have many different goals, but for this first goal, for the mutational signatures, the study designs aims to collect specimens from a few hundred subjects with a high levels of exposure to known risk factors for lung cancer in never smokers, like radon, coal, air pollutions, and a much larger number of samples from the general population with a known etiology. And we are conducting whole genome sequencing in frozen tissues and also transcriptomic and epigenomic analysis. I will first explain a few results that we obtained in the first set of 232 cases that we analyzed and recently published, and it contained, included the 189 adenocarcinoma, 36 carcinoids, and 7 mixed types. And I will also illustrate more analysis in mutational signatures in a larger data set, including non-smokers and smokers. The first thing we observed when we looked at the tumor mutational burden in the first set of 232 cases is that in this Sherlock study in the subjects with the non-smokers, the mutational burden was lower, and particularly in the carcinoid subtype, than many other cancer types, including adenocarcinoma from smokers that we obtained from the TCGA study. Now, using this low mutational burden, we then extracted the mutational signatures. And at the time, we used the SIGA profilers with the 1,536 mutation types. We used these mutation types because we were hoping to distinguish better the mutational signatures given the low mutation burden. And this is the landscape that we observed. Every column defines a different tissue sample, and the colors define the signatures. So I highlighted a few of them, like the signature 3, potentially associated with the homologous recombination deficiency, the signature 18, associated with the reactive oxygen species, and the ApoBec, and a few others. But as you can see, there are many samples that are dominated by signature 40 or 8 or signature 5 that are considered still of unknown origin. And so we wanted to see whether the endogenous processes versus the exogenous processes can explain better these mutational patterns if we exclude the unknown signatures. So we looked at the entire cosmic signatures and then extracted those with endogenous processes. And we also tested the environmental mutagen signatures that work from serina in pluripotent stem cells and the combination between endogenous and exogenous. But as you can see, the cosine similarities of the endogenous processes seems to be the best. And in fact, when we compare exogenous versus endogenous, all samples were actually better explained by the endogenous processes in these samples than exogenous exposures. And this was true also for 62 tumors that derived from subjects that reported secondhand tobacco smoking exposure. We did not see any signature 4 in these samples. In fact, when we compare the patterns of mutations in never smokers with passive smoking versus never smokers without passive smoking for single base substitution, double base substitutions, and indels, we didn't see any major difference. Cosine similarity was almost 1. So we tested whether this could be due to misclassification by using only samples where we could really measure exposure during childhood, at work, with the spouse, and we created a scores with high and low levels. But even when we compare the highest with the low levels, granted in a very few samples, we couldn't see any major difference. So we went to simulations approaches to try to understand if these results were due to a detection limit of our algorithms. So we injected mutations associated with SPS4 at 1, 5, 10, 15, and 20% in 62 tumors. And we saw that in order to be captured by our algorithms, these mutations had to be at least 15% in these 62 samples. So if there were mutations associated with the secondhand tobacco smoking in these samples, they had to be below this threshold. Now, these results were based on the first 232 set of samples. We are now analyzing over 1,200 tumor normal pair whole genome sequencing data and over 1,800 with RNA sequencing and methylation data, also including some public databases. So currently, we also included 30% of Eastern Asians, and in the future, we will have also Africans, Caribbean subjects, and also from the Middle East and Latin America. But in this analysis, we're comparing European and East Asian. The larger majority of the histologies is adenocarcinoma. Females are 64%. And we also conducted a whole genome sequencing in a group of tumors from smokers, so that we could compare the landscape between smokers and non-smokers, taking advantage of the same platform, the sequencing depth, analytical pipelines, and so on. So first we extracted the de novo signatures. We identified approximately 14 with the 0.9 average stability. And then we assigned the signatures and we used the seq profiler and tested the different mutation types combinations. So we tested 96, 288, and the 1536. But we saw major differences across them. So for example, if we use the 1536 that we used in the first set, we saw that there were quite a large percentage of the samples that had the mutation on the signature 40, which was totally absent when we looked at the 96 combinations. In the 96 combinations, there was a signature 8. It was absent in the 1536 combination types. When we looked at the 288, we saw that signature 40 and signature 8 were also present, although in a smaller percentage. But in this, we also identified a few samples that had the signature 3, this potentially associated with homologous recombination. And so we thought that this combination of mutations capture better the landscape, also because it takes into consideration the strand bias, which is very important for the signature 4, the signature associated with tobacco smoking. And given that we are studying lung cancer, this was crucial. So we decided to use the 288 mutation types, but we kept in mind that there is very large difference about just changing the mutation types. And this not only when we looked at different mutation types in the seq profilers, but there are many differences also across algorithms. So for example, when we compare seq profiler with signal, we saw that, for example, the clock-like mutation signature or the ApoBec were captured better by the seq profiler, or at least that there were more mutations assigned to these signatures in seq profiler than in signal. And vice versa, if we looked at the signature 8 or signature 18, there were many more mutations associated with these signatures according to signal than according to seq profiler. And similarly for other signatures. Even signature 4 had some discrepancy across the algorithms, but much less. And we were happy about this because we are using a lot of the signature 4. So among some of the differences we saw when we compared the European the non-smokers versus the smokers, as expected, larger number of mutations were associated with the signature 4 in the smokers. And in the non-smokers, we saw the signature 8 and signature 18. That was also present in the Asian samples. But in the Asian samples, we also found the signature 22, the signature that is associated to aristolochic acid that Peter showed this morning in hepatocellular carcinoma, and had been also found in renal cancer, in Balkan areas in Europe. And these are all associated with this plant that in this area of Taiwan, when we collected these samples, is used as a sort of Chinese herbal medicine. We also had a small number of samples, but very interesting samples from the Shanghai area in China, where the people are exposed to very high level of coal combustions. And in fact, the never smokers in this area have a risk of lung cancer that is much higher than the risk attributed to tobacco smoking. And what we saw that actually all of these coal combustions, was associated with the very high level of the signature 4. 84% of the mutations were actually attributed to this signature 4. So this is interesting and corresponds to what we see epidemiologically. And in terms of the driver genes, just to give an idea, so we have different patterns according to the prominence of different driver genes. In KRAS, TP53 is more flat, but reflects a lot of the tobacco smoking. EGFR in non-smokers has a typical hotspot like the KRAS. Then we wanted to verify whether we could see any signature 4 in subjects exposed to secondhand tobacco smoking, since in the first set, we didn't see any. So in this larger set, there were 250 subjects among the never smokers that reported to have various degrees of secondhand tobacco smoking exposures. But we saw that the 246 out of 250, so practically over 98%, did not have any signature 4. So again, this confirms our previous findings that they could be related to detection limit of the algorithm, could be detected that related to misclassifications, can also be due to the fact that the secondhand tobacco smoking may generate a different type of signatures in these samples, but can also reflect what we know from epidemiological observation, that secondhand tobacco smoking is a weaker carcinogen in comparison to the active smoking. If you know the relative risk of lung cancer associated with secondhand tobacco smoking is 10 to 20% higher than never smokers, but much lower than the 15 to 25-fold risk associated with active smokers. Now, given that we also had this smoking cancer, we wanted to test how the signature captures different levels of exposure to tobacco smoking. So we relied on over 200 cancers from smokers, and they were quite well distributed between current smokers and former smokers. And we had very detailed smoking exposure information and very balanced grouping according to different variables like age of first beginning smoking, cumulative pack years, that is the combination between intensity and durations in years, and years since quitting smoking. And what we saw is that when we looked at the SBS4, DBS2, and the ID3, the typical signatures associated with tobacco smoking, they captured the increase in cumulative pack years quite nicely. And similarly, there was a decrease with increasing number of years since quitting smoking. But instead, there was no major effects, apparently, of the age at first cigarettes in these samples. This was a very nice confirmation that this signature can capture the long-term exposures. And we didn't see the similar patterns when we looked at the different type of signature, the clock-like or the ApoBac. We also wanted to test in an orthogonal way the signature tree. As Daniela just mentioned, when you have a signature tree, particularly because it can have clinical implication for suggestion of treatment, you really need to verify this. Also because this is a typical flat signature. We had between 14 and 32 samples in Asian and European, respectively, when we saw in this analysis. So we used the HR Detect to try to see if we can confirm this finding. And we saw there were only 8 samples here that were identified. They were all non-smokers based on this algorithm. And when we tried the CORD, the random forest classifier of homologous recombination deficiency that was trained across different cancer types in the PICOT study, we saw more samples that were associated with homologous recombination deficiency. All were never smokers. And there were 6 and 17 BRCA1 and BRCA2 types. Some of this could be explained by germline variations in the BRCA1 and 2 genes and other genes in the HRD pathway, but not all. So there were some discrepancy, and even considering some somatic mutations in these genes. And so when we compare then the CORD versus the HR Detect and the SBS3, we saw that all the samples that were identified as high HRD by the HR Detect were also identified by the CORD, and 7 out of 8 samples also had the signature tree, here identified with the triangle. But this signature tree was also found in many samples that were not defined as high recombination deficiency by CORD or HR Detect. Again, emphasizing the need to do multiple approaches to verify these identifications of this signature, particularly if this is used for patient stratifications and suggestion for PARP inhibitors or other treatments. We also looked at the cluster mutations, and we saw between 1 and 5 different clusters per samples, and they were mostly clonal. And then, as expected in the never smokers, the mutation signatures in these cluster mutations reflected the effect of the ApoBac. But in the smokers, the cluster mutations also had particular signatures that resembled the smoking exposures. And so clearly the smoking can also contribute to the formations of these cluster mutations. There are many other analyses that we are conducting, including copy number alterations, complete genomic rearrangement, exposures to other signatures, and also other exposure for radon and other important risk factors for lung cancer. We are also testing whether there are different signatures based on specific cells of origin, and whether microbiome in the lung tissue can affect these signatures. Now, in the last couple of minutes, I just wanted to report on an effort that we're making, and particularly Tonggu Zhang in our team, to create a sort of a portal or repository for mutational signatures, MSIG portal, that we hope to be able to launch in the next few weeks. And currently, this portal includes 4 modules: catalog, visualization, explorations, and signature association. In terms of the catalog, we are listing a lot of signatures. We have the genomes of human and mouse, different genome builds, different versions of the cosmic signatures, the cancer-specific signatures, environmental mutagen signatures, and so on. And through the signature visualizations, we currently uploaded all the whole exome sequencing from TCGA and whole genome sequencing from all the available studies in the literature, but one can also use his own or her own data and conduct some analysis comparing the mutational patterns at the sample level. For example, testing the profile summary, the typical mutational profiles, or comparing different profiles, using cosine similarities, principal component analysis, even looking at enrichment analysis, and so on. And the signature exploration, again, one can use his own data or using cosmic, mutograph, organ-specific cancer data, and test for the activities of each mutational signature in the samples. And so, for example, looking at the prevalence of the mutations or in each samples or the mutational signature, combinations at each sample level, or associations across signatures, or the landscape in the samples of the signatures, and so on. We also uploaded a lot of data for the signature association, clinical data and structural variants, copy number, mutational burden, MSI counts, telomere, and many other features. So one can use this to conduct univariate or multivariate analysis, adjusting for each other, and testing association of mutation signature with purity or other factors like age or sex and many other factors. And finally, there is a list of publications that we reported in the portal, not only those from which we derived all the signatures, but also reviews to have the state of the art on mutational signatures and the different computational approaches that are currently available. And again, we hope to be able to launch this in a few weeks for the research community and gather results from them and see whether this is useful and should be maintained. And finally, I wouldn't be able to show any of these results without a fantastic team and their supportive families and many, many other investigators, including some that are present here in this symposium. Thank you.
Thank you very much, Thierry, for excellent presentation. We have a couple of questions in the chat as well as in the question boxes. I'll start with the chat. Is SBS40 distinct from SBS5?
Very, very good question. So this is the problem that we have. So again, if we use the combination of 15 and the 36 mutation types, we can distinguish the signature 40, but in other combinations of mutation, not. And this is the issue of signature 5, 40, and 3, the flat signatures. In fact, in some of our analysis at the end, we put them together because until we have the possibility to truly distinguish them, this remains an open question.
Okay, another question is, there was 3 hypermutators on the figure. Did you find a cause? This is one of the earlier figures you had.
Yes, actually there were 4 hypermutator, and they were strongly associated with ApoPEC.
Okay, thank you.
3 of them were non-smokers, by the way, and 1 was smoker.
Okay, I think we have time for one more question. Will the signature portal also contain list of mutation calls used to generate signatures?
Sorry, could you repeat the question?
Will the signature portal also contain lists of mutation calls used to generate signatures? This is from Dr. Gordani.
Maybe Dimitri, could you ask directly?
Yeah, he perhaps, yeah.
Yes, can you hear me?
Yes.
So in order to generate signatures, you use some program, some mathematical tool, and mutation calls. In order to access these mutation calls with other tools, someone would need mutation calls. Will your portal contain mutation calls that were used to generate these signatures? And I believe this is the common problem for scientists who want to follow up signature research.
Yes, I totally understand your questions, and these very important questions. And yes, for now, there are all the mutational patterns, mutational calls that will allow you to do all the analysis of the patterns. Now, in the future, if we are to maintain this portal, we have to discuss how to collect this type of information. Yeah.
Okay, I think we need to move on. Thanks again, Thierry, for your presentation. We're now moving to our last presentation in this session, Dr. Gadi Getz from the Broad Institute, and the title of his presentation is Mismatch Repair and Polymerase Proofreading Deficiencies in Tumor and Normal Cells. Dr. Getz.
Sorry, thank you. Can you see my slides?
Yes, very good.
Great, thank you. Thanks the organizers for inviting me to share kind of our experience on mutational signature. In particular, I would like to talk about mismatch repair and polymerase proofreading deficiencies in tumors and normal cells. So here are my disclosures. The most relevant are patent applications related to MS-MUTSIG, MS-DTEC, and MS-SIGs. So the outline of the talk is background on discovery of mutational signatures, and then 2 more recent stories about detections of tumors with microsatellite instability from cell-free DNA, and the other one is mutational signatures in constitutional mismatch repair deficiency or deficient tumors or patients in tumors and normal cells. So discovery of mutational signatures. So my group started to get interested in mutational signatures in the process of looking for drivers. Because when we look for drivers in the genome, we look across patients, and then we kind of count how many mutations we see per gene, but we know the different mutations and different types of mutations occur at different rates, and we need to take that into account to see how much we're really surprised by the number of mutations we see in a gene. So when we looked at mutations across cancer, this is whole exome data, we already saw that the types of substitutions are different, with a very clear kind of C2T mutations in the UV light and the C2A in the cigarette smoking related mutations. When we look at it then in not only the mutated base, but the base before and after, this is the 96 kind of classes of mutations. And we often like to also look at it in this kind of what we call the Lego plots, where you can see the different colors or the different types of mutations, and each 4x4 quadrant here or area here represent the base that is before and after. And this is kind of the signature 1 CpG2T. You see that all of these are C2T yellow mutations, followed by G in this kind of last column here. So when we looked in cancer, that was kind of early on, in different cancer types, we already noticed that there's some patterns that are similar in different cancers, but are not in other cancers, and they're in different levels of activity in different cancer types. So then we came with this model that can we represent the mutations we see in a sample as a sum of different signatures, and can we learn those signatures from the data? And like similarly done at in parallel at the Sanger Institute, we used non-negative matrix factorization, which is an algorithm developed originally to look at faces, images of faces, to find different features in the faces and represent the activity or the level of each feature to be able to reconstruct the face. In our case, we have the mutational spectra across samples. We want to find or learn mutational signatures and the actual activity of each signature in every patient, that such that it could reconstruct the spectra of mutations. So all the observed data are non-negative weighted sums of these components, and that's the reason it's non-negative matrix factorization. So the way it works, just for most of you kind of know that, but this is the matrix of, let's say, 96 classes of mutations across all the samples, and we want to approximate this matrix by a product of two simpler matrices, one representing in the columns the signatures, and the other one, or the columns of the other one, represent the activities of all the signatures in a particular patient. And what we don't know, however, is how many signatures we expect in our data. And this is something that we actually need to feed to the algorithm and play with it. But, and that's a big limitation of non-negative matrix factorization. So we used 3,000 samples back then to find, then we found 6 signatures. You see the Applebeck, you see the CPG2T, you see the smoking signature, you see the UV signature. This was actually a sequencing artifact signature, and all the rest were kind of, we call them miscellaneous. At the same time, the Ludmil and the Sanger team studied 7,000 tumors and came up with 21 signatures, which was later became the 30 signatures in the version 2 of the COSMIC database. So then we asked ourselves, well, how can we actually learn this number of signatures from the data? So Jay Gil Kim in the lab used a version of NMF, which is called the automatic relevance determination version of NMF, which is a Bayesian type of method that uses priors on the values of these matrices, this signature matrix and the activity matrices, that is an exponential form, and which means that the majority of the mutations are 0, but there some of them are higher, and that is that prior on this on each of the signatures of this exponential form has a parameter for every signature, which is called the weight of that parameter. And as you iterate and learn fit the data, you learn these parameters from the data, and some of the weights converge to 0, and therefore we remove them. So we start with a larger number of candidate signatures. We started randomly, and then we converge to a smaller number of signatures given the data. So we don't need to give the number of signatures to the algorithm. It gives it back to us. But then, since NMF and both this automatic relevant determination NMF, they depend on the initial conditions, and you could run them many times and get different results. So we actually run it multiple times and generate something that you could think of as a posterior distribution of the number of signatures, and we choose the most likely K given the data. And we call this algorithm and this process, this algorithm is called Signature Analyzer, and it's available. So then we joined as part of the pan-cancer analysis of whole genomes. We joined forces with Mike Stratton and Steve Rosen and Ludmil, Jay Gil and Nika from my lab. And I'm sorry, I don't have a photo of Mini Wang from Steve Rosen's lab. And we analyzed all the whole genomes and exomes that were available at that time with both the SIG profiler and signature analyzer. And we found 49 signatures of single base substitutions and 17 indel-related signatures and 11 double base signatures. We got similar results by applying the two different algorithms, but not the same. And particularly, they were different in the flat light signatures. So when we heard earlier today of several talks about these flat light signatures and can we find them or not, I urge you to actually try multiple methods and see what they come up with in terms of these flat light signatures. In addition, we also heard just now about which types of classes of mutations. Should we use the 96? Should we use 192 or 1536, etc.? And there's different double strand and double base substitutions or indels. We prefer, if we have a lot of data, to use the composite signatures, which are the 1536 plus the double base substitutions and the indels as one kind of vector to represent all the different mutations, and then learn them. If we have a lot of whole genome data, learn them from the data. But that only works when you have enough mutations, because otherwise the matrix is too sparse and cannot learn the signatures. Then, since we need to run it many times, and even running it a single time, these algorithms take a while. So we wanted to improve the running time of these algorithms, and we leveraged GPUs, graphical processing units, and we were able to implement Signature Analyzer on GPUs that could run 200 times faster, and that is available and published and available through our GitHub repository. And you could run it many times, and it could scale linearly, and you could run hundreds of thousands of samples in minutes using this GPU sped up method. So that was kind of the introduction. So now we'll talk about two different projects. One is detection of MSI cancer from cell-free DNA. It's a yet unpublished story. So this was led by Yossi Maruvka, a postdoc in my lab that now started his own lab. So as you all know, the FDA approved pembrolizumab as a PD-1 inhibitor to treat any solid tumor that is MSI high, meaning that it has a mismatch repair deficiency. So what we ask ourselves is, can we identify easily MSI cancers from cell-free DNA from a simple blood draw? And that would kind of enable patients to be treated with these PD-1 inhibitors. So as you all know, the mismatch repair pathway identify both mismatched bases, but also when there are actually microsatellites and there are loops, and it could identify those loops and eventually kind of repair both the bases and loops. So a dysfunction of one of these MMR genes leads to an elevated mutation rate of single base substitutions, or SNVs, and indels, and particularly indels in microsatellites. So just to make sure that everyone is on the same page on microsatellites, microsatellites are repetitive regions of the genome that they have repeats of small motifs. For example, here there's AC, 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 AC. Most of them are just a repeat of one nucleotide, like AAAA or CC, CCC, etc. And there are roughly 23 million microsatellites in the genome, and 300,000 or 400,000 in the exome. So due to this DNA slippage, I think there's a cartoon here. When DNA is replicated, the polymerase can kind of detach and attach again. And if there's a microsatellite, loops could form. And then you could either increase or decrease the number of repeats in these microsatellites. And the mutation rate in these microsatellites are much higher than any other part of the genome, with 1,000 to 100,000 more mutations than in other parts of the genome. Typically, they're caused by, in cancer, by hypermethylation of a promoter of MLH1. There's a syndrome called Lynch syndrome that is caused by heterozygous germline deleterious mutations in either MLH1, MSH2, and then flow frequencies of MHH6 and PMS2. And later in life, a second hit could hit the wild type copy, and then that increases the chance of getting cancer, particularly colon cancer and endometrial cancers, stomach cancer. The slippage happens in all the cells in the body. However, they're not being repaired as efficiently if the mismatch repair mechanism is dysfunctional. So how do you find them? How do you find MSI in the clinic today? Typically, you perform immunohistochemistry of the mismatch repair gene, for example, MLH1. And if you see that you don't find it, that means that you're potentially a microsatellite unstable. The more common way of, or another common way of doing it is using kind of gel-based assay that looks at a few microsatellites, actually 7 microsatellites, what's called the Bethesda protocol, and looking for indels in them. And if you have 2 or more indels in those 7 sites, and typically 3 base pair indels, then you call it an MSI high tumor. And now there's, of course, next generation based sequencing method that deep sequence MSI loci to find microsatellites that are mutated. It's not easy calling mutational indels in these microsatellites, because when you look at a particular site and you look at the reads that are covering those sites, because of the DNA amplification in the sequencing process itself, you get different length of microsatellite indels. You see it both in the tumor and the normal. So it's very difficult. It's not as direct as calling somatic, regular somatic mutations, because you need to compare those 2 distributions and figure out that this is actually different from that. And that's not as easy. So we developed in the past a method called MS-Mutect that calculates essentially a type specific, meaning for every type size of microsatellite, a noise model, a distribution of what are the likely indels that you see in those microsatellites in normal tissue or in clonal tissue, but without mutations, I mean. And then using these noise distributions, we could actually calculate what we see in the tumor just a noisy version of what we saw in the normal, or is it a true somatic mutation in the tumor? And we could call somatic indels in these deep sequenced microsatellites. And we validated using RNA sequencing data. But now we wanted to look at cell-free DNA. And so we've developed the Broad, together with Victor Stenson, Gavin Hahn, Sam Freeman, Todd Golub, and Chris Love, and Matthew Marson, approach to look at whole exome sequencing of cell-free DNA by extracting the library of cell-free DNA and performing, looking at copy number changes throughout in the cell-free DNA, and that could detect if there's tumor DNA in the cell-free DNA. And the sensitivity of that is roughly the lowest sensitivity of 3%. So if you have less than 3% tumor DNA in the cell-free DNA, we can't really say that there's tumor in that cell-free DNA. So the second part that I want to talk about is mutational signatures in constitutional mismatch repair deficient patients, or CMMRD. So there are patients that unfortunately inherit 2 deficient or defective copies of a mismatch repair gene. And those kids unfortunately developed multiple cancers at very early age. And this is a work done together in collaboration with Uri Tabori from the University of Toronto and Brian Chung from his lab and Yossi Maruvka. And so we looked at these patients, and if you apply the regular MSI detection tool in those patients, in their normal cells, you find no MSI high. And even in the tumors, you find most of them are called MSS, as they're not detected as MSI tumors, although every cell in their body has no mismatch repair. And what's going on here is that you essentially don't see mutations in the canonical 5 sites of this Bethesda protocol. And in fact, it actually is different. And what we saw here is that even in a single patient in 2 different sites, it could identify 2 different kind of mutations, but not enough to call it MSI high. And here you see that actually in different parts of the tumors in different parts of the body have different frequency of being called MSI high.
Dr. Getz, time is over. Okay,
so where are the MS indels? Let me just say here that by using MS-Mutect, we were able to find MSI microsatellite in each of these cancers. So it's just the Bethesda protocol, and sorry but we also show now that we could do this similar to MSI Detect using ultra low pass whole genome sequencing. And the most important thing that because this tool that we have does not need the mutations to be clonal, we could detect even from blood that these patients are CMMRD, and that has a very important clinical implication because some of these kids they have non-canonical mutations in those genes, and they don't know that they are CMMRD patients. And using just a regular blood test from normal cells without even a tumor, you could detect that that kid is a CMMRD. Yeah, I will just finally say here that we could see that these signatures that we found could actually distinguish between patients that respond or don't respond to immune checkpoint blockade. And one last point I want to say is that the mutations in these adult mismatch repair patients in microsatellites seem to converge on 15 bases. So if the original size of the microsatellite was more than 15 bases, it actually has deletions that bring it to 15 bases. And what we think is going on here, the model, is actually that reflects the size of the polymerase, that once it's in 15 bases into the microsatellites and it flops up and down, it could then jump to the other side of the microsatellites, and it actually prefers to have 15 bases of a microsatellite as cells divide. So just to summarize, I told you about signature analyzer as this Bayesian version of NMF. We actually see that in some cases, the linear assumption of NMF is incorrect, and I urge you to read this paper. And I talked about MSI detect to be able to detect MSI from blood from ultra low pass whole genome sequencing, and that we could detect these microsatellite signatures and distinguish a CMMRD patient also from both their tumors and their normal cells. And both of these results have direct clinical implications. And I want to thank the many people involved in these projects, Jay Gilkim and Nick Cardvala, Amara Taylor and Francois Arguet and Yossi Maruvka, the collaboration with Uri Tavori and Brian Chung, and the collaboration with through the PCOG with Mike Stratton, Ludmila Ksarnov, Steve Rosen, and Minnie Wang, and the entire PCOG. I'll be happy to take questions.
Thank you very much, Dr. Getz, for the interesting presentation. We have time to answer a couple of questions. I have a question here from Terry. Can you identify cell-free DNA for mismatch repair deficiency testing in early stage cancers?
We are working exactly on that. That's our goal, is to try to use that as early detection for cancer of MSI cancers. And both, yeah, that's our goal, and to monitor Lynch syndrome patients. Thank you.
The other question is there a published tool or workflow that provides composite 1536 SBS signatures plus DBS and indels as one vector for each sample?
I don't know if there's a tool to do that. It's quite easy to assemble this from the existing, but I can. Please send me an email and I will find the tool to do it, because we are doing it all the time. So I'm sure we have code to do that. I don't know if it's part of our tool or we'll be happy to make it available.
Mm-hmm. And one final question for the analysis of exomes, what signature tools would be convenient to use? Is Seq Profiler designed only for WGS or also for exomes?
So Seq Profile is Ludmil's method, so I'll have him respond to that. But Signature Analyzer could work easily on exomes and genomes. We worked for many years on exomes with it, but then I wouldn't advise using the 1536. Because of just the number of mutations is too small, I would just use the 96 mutation kind of classes. Because if the matrix is too sparse, it's very difficult to learn the signatures because every patient is different.
Okay, thank you very much. So I think this is all the questions that we can take for now. Because of the time, we need to move to the panel discussion. We have concluded the 4 presentations for the cancer and normal tissue session. And now I invite Dr. Nuria Lopez Vegas from IRB Barcelona to lead the discussion, and also invite the 4 speakers as well as Dr. Peter Campbell to join with Dr. Lopez. Dr. Lopez, I'll hand you the microphone.
Thank you, Professor Yassin. And I think we have here all the speakers. So great talks. We have now 20 minutes this is normal and cancer tissues section. So I prepared a few questions, and I would like to start by differences in ethnicity. So I think that was touched upon by Daniela, but also by Tere. And I would like to see, if, from your opinion, how many surprises are probably still expecting us to find by the fact that we have only analyzed very limited parts of the world. I know that Phil Jones says that he doesn't like the term ethnicity. Maybe we should use different genetic background. But it's true, we have analyzed only a part of the world, and there is very important biases. And here there are two questions. How many surprises do we think we still have to find? How many differences we may find in the mutational signatures, in the mutation burden, and also in the drivers? And the second question is, how should we do it at international level so that we can overcome the challenges and see if we can have a broader view of cancer genomes and mutational signatures across ethnicities. I don't know who wants to start.
Well, can I just say quickly, because it's something we're thinking about. So I think when you look across germline, but I don't like ethnicity because it's loaded. Okay, so if we just think about different germline populations, and there are enormous variations in cancer incidence, and there could be environment or germline, obviously. And it's very interesting if you look at the Nature Genetics paper from the Mutagraphs project, Moody et al., that Mike Stratton published a couple of weeks ago, which was a lovely study, and it ought to have found beautiful mutational signature differences in esophageal squamous cancer, where there were huge variations in incidence in hotspots across the world, and it didn't. And I think that's sort of a very well executed study, and it tells us something. And then our little silly pilot study found big differences, but is fatally confounded by the fact that we're looking at two populations thousands of miles apart. So what you want to do if you want to look at these things, and countries like America, National Cancer Institute, you have populations in a single state who are ethnically, if I can use that word, germline, genotypically diverse in very similar environmental locales. And I suspect if you look at things like skin cancer, they will have huge differences in the rates. If you look around the world at keratinocyte cancers, which I do know about, the differences are massive. And you could study that, and you will find other cancers where there are massive differences. And you've got it in America because you've got diversity and within a confined locality. And you just need to get on and do it. It's a very obvious thing to do. It's for the rest of us, it's with more homogeneous populations, it's much harder. I want to talk to people. Actually, somebody reached out to me through the chat just now from the US. I'm very excited about that. But that it's a huge opportunity for you guys to do that, because if you start moving geography, it gets much more complicated. There you're kind of playing with the environment dial as well as the genotype dial. I'll shut up.
Tere?
Yeah, these are all very important questions. And yes, I was puzzled by the results of the Mutograph originally, because I was personally expecting more differences, like reflecting the different incidents. In our experience in the Sherlock study, for now, based on subjects of European descent or East Asian descent, with germline confirmation, the major difference, for every type of genomic changes, we saw that if we compare the European smokers versus Asian versus European non-smoker, we see that the Asian have more richness of any type of genomic changes we saw, like structural variants, copy number alterations, mutations in never smokers from Asia than in never smokers from Europe. It's like the Asian population is in the middle, the never smokers in the middle between the European non-smokers and European smokers. So we were puzzled by this, and we're still, conducting a lot of this analysis. But we saw also that there are different frequency of the drivers and the frequency of the hotspot mutations in the drivers across population. For example, in the EGFR, it's much more common in Asian population, and the typical hotspot in this EGFR is different in terms of frequency than the hotspot that is much more frequent in the European population, just to give you an example. And also I wanted to say one thing about what Phil was suggesting, do this in the US. Well, we're trying to collect all these samples, but I wanted to also emphasize the fact that it is not easy. There are some populations that are less enthusiastic, let's say, in participating in research activities. And so to collect samples from different ethnic groups, call it ethnic or call it germline difference, is not as easy as collecting, European samples. So there is that issue also to consider.
Mm-hmm. And Daniela.
No, I completely agree. And also, well, for example, here in Latin America and Mexico, where I am based, well, the population is not homogeneous at all. Like, we're all called Latino in a way, but there's people in the north of the country that look completely European. There's people in the south that just look completely different to that, and they have very different genetic germline variants, right? So it's also, and their environments are also very different, because at least in Mexico, the north is generally more affluent than the south. So it's not only a germline genetic difference, but also an environmental difference, which is quite difficult to dissect. So in the map I showed you in my talk, is like, well, most of our patients come from the south. We think it's just because it's close to the hospital, but if there's something else there about genetics, we may not be able to see it because of these problems. But I agree. I do think that they will be some differences, maybe not as much as we thought before, but it's important to find them for applying directly those results in our populations. Now, the second question that Nuria asked, and I thought was very interesting, is how should we do these projects? Is it at an international level or at a more local level? I would be tempted to say, of course, international, because this is, a big collaboration with, let's uniformize the methods that we're using, and, the same benchmark, and hopefully, make results comparable. However, there is also, for local researchers, a problem there where sometimes there's not a leadership role for, people that are basically not in the global north. So this makes it also a bit harder to collaborate just just like that as an international consortium. So I think there must be, I would like to join, for example, a consortium like this, or make it international, but I think there must be very clear consideration for not only the samples of these other countries, but also the local expertise that the scientists have, and that they know their populations, right? So, not just contributors, but also a leading role in those projects. My point of view.
I fully agree with that. Let's call for a kind of international collaboration that is, as you described, not just getting samples and sequencing them all in one country, but in a way that it also encourages for this distribution of expertise around. Okay, I'm going to change topic now because this is the section on normal tissue as well, and I'm having here also Peter, John, and Gadi may also have an opinion on that. So mutational signatures in the normal tissue, and in particular, I know it's about cancer as well, but what about mutational signatures in aging? So mutational signatures and in general somatic mutations, and what's their role in other diseases that are not cancer. Peter presented about liver diseases. There are interesting results about inflammatory bowel disease in colorectal, but for example, neurological diseases or cardiovascular problems, do you think that somatic mutations may also play a role there, and how we should study that?
Perhaps if I start, Nuri, I think that's a really interesting question. Essentially, we don't know what role that somatic mutations may play in diseases other than cancer. As you say, we have, the data I presented today on liver, and there's data in inflammatory bowel disease showing that mutations are clearly being selected for in these different diseases. It's a more complex inference process in many ways than looking at inferring cancer genes, because selection is the kind of clonal evolution and clonal proliferation in that sort of cell autonomous way is the defining feature of cancer in many ways. But in diseases other than cancer, where many of the disease processes are not kind of driven by a single cell type, and there's often interplay between different cell types, and sometimes there's a kind of, a war between cell types. So in, for example, autoimmune disease, where there's effector cells in the form of lymphocytes and inflammatory cells, and then affected cells in terms of the kind of target tissue. It's not clear, what the significance is of mutations in either the effector cells or the affected cells, and we're beginning to sort of wrestle with that as we look at these inflammatory diseases in more details. The sort of data that's emerging suggests that the landscape of selection changes in many diseases. So the sets of mutations that confer a selective advantage are different. What we can imagine, if we take, for example, just the one instance of the FOXO1 mutations that I described in the liver disease today, there's two ways of looking at that, and it's kind of interesting to imagine, would this be an interesting therapeutic target? And you could imagine a world in which you might want to mimic the effects of the FOXO1 mutations, because clearly the mutations are protecting the hepatocytes against the disease process. Those hepatocytes are surviving better in the toxic microenvironment of that liver disease. And so it might be a way of sparing liver function. Also, those mutations are not seen in liver cancers, so it may well, by affecting that function, one may well slow the progression down to liver cancer. On the other hand, it may well be that these mutations, in kind of protecting the individual clone, do so by disrupting the function of that clone. And so maybe that hepatocyte clone can no longer fulfill the functions of a hepatocyte. And in particular, given that FOXO1 is the master regulator of insulin signaling, maybe that loss of function actually contributes to the insulin resistance of, for example, type 2 diabetes. And so if one had a drug then that mimics the effect of FOXO1, actually maybe you might make those metabolic phenotypes worse. So I think it's really, it's a more complicated landscape than we imagine in cancer. Having said that, it may be also quite complicated in cancer. The data that Phil presented suggesting that Notch1 mutations actually, although they're seen in cancer, may actually slow progression down. and be in some sense beneficial for the host. Maybe we're underestimating the complexity of selection in cancer, in assuming that everything that we see under positive selection is a valid therapeutic target.
Mm-hmm. Other comments on this topic? I have other questions. So, okay. So I was puzzled by some of the results showing that clones that are very nearby may have different mutational signatures. And I want to pose a question. It's a more technical question because identifying the small clones and seeing that it already lead us to very interesting results. Imagine the day that we could see at single cell level, that we could see whole genomes at single cell level. We're looking at the future now and be able to see the differences in the mutation rates, mutational signatures. How far are we from that?
Yeah. Well, technically, we're quite a long way away from that. I think what we have, what there are protocols to do are to light up. So one can do at the moment. One can do base specific. So one can do spatial transcriptomics, and in theory, one can call mutations from those single cell transcriptomes. Either single cell transcriptomes or spatial transcriptomes. It's rather more complicated than calling mutations from nice bulk genomic DNA, but people are beginning to look at this. There are also ways that one can light up transcripts in a base specific way in tissues. So if you know the mutations you're looking for, then that's fine. I think there's nothing. The way that these spatial transcriptomics methods work, it's not impossible to imagine a world in which you digest the genomic DNA in situ and then elute it off the tissue section and then turn it into a sequencing library and a barcoded sequencing library where the barcodes represent a spatial, postcode essentially.
You're talking about DNA here, Peter. DNA?
DNA, yes. I wonder whether I don't know. I hope that people are working on this, but that would be amazing because you could. I reckon you're probably not going to get down to single cell resolution, but you could certainly get down to clones of 10 cell resolution with that, and I think that could be, those kind of methods could be really interesting. But I'm totally speculating. And I have no idea whether anyone is working on such protocols, but I think it's not impossible to imagine a world in which we could do that. And then, as you say, imagine the experiments you could do if you could crack that one.
Yeah. Let me just comment here that I think that, like Phil said earlier, looking for the ApoBec. We know ApoBec behaves in a bursty manner and not in all the cells, but looking at ApoBec kind of clones and where they are and what happens with them, I think would be good with this kind of future technology that we are envisioning here. Because I think you need to look at many cells in order to find them, because it could be a rare event, but still local and might be exciting to study those things. So I agree that it's a good direction.
Okay. Any other comment on this topic? We just have 1 more minute, so I'm not going to touch any other question.
Well, I think looking for ApoBec we've burnt hundreds of genomes in normal esophagus, and we found it in 1. And it wasn't anything like as spectacular as it is in a tumor. So I think I agree, it's a bursty, quick, annoyingly rare thing. And you have to remember that there are so many, it's one of these, cancer is so rare, all of these somatic mutations and in your esophagus, and your lifetime incidence is 1%. So these mutations don't know that they're cancer mutations. They're just stem cell regulatory genes that are messing up clonal fate. And the cancer transformation event is quick and astonishingly rare. And it'd be lovely if there was a pre-transformation signature, but I'm getting the feeling from mutagraphs, etc., that there really isn't. It's quick, savage, and blink and it's gone. And you're already there.
Okay, so time is over. I would spend many more minutes and hours discussing with you, but we have to finish here and move to the next session. Thank you.
Thank you.
Thank you so much, Dr. Lopez-Picas. I think it was a very rich discussion. I'd like to thank you and thank all the presenters. We had a great morning session and was very educational for everyone.